Hello, my name is Marilyn Jimenez. I am a junior here at Aruleta University. My presentation is on temperature-dependent sex determination in turtle species, which is also referred to as TSD. Um, I decided to do my research on this topic because when I was brainstorming the type of research I wanted to do and the animals that I wanted to do my research on, I knew that it was either canines or turtles, but I didn't know what exactly. I knew that it was something that I didn't know about. So when I talked to my professor, he did mention that there were crocodiles that were only born males in that area, which was bad for like reproduction. They didn't have females to reproduce with. So I thought that was really interesting, and I did my research on that, and I found out that it had a lot to do with the temperature that they were um like where their eggs were being incubated in, and that it could also affect turtles. So I thought that was a perfect thing, a perfect topic to research about, and it included turtles. And I thought that was it was a little bit ignorant of me to not know that you know, turtles, I knew that they had nests and that they had to incubate, but I did not know that their sex determined on the temperature. Like I had no idea. And it makes you think and wonder, like, about climate change and stuff like that, and if that has a an effect on turtles and stuff like that. So yeah. So first we're going to be talking about my outline, which is just a little background of what this presentation is like, what the slides are going to be about. I wanted to start off with like background information. When I'm reading an article or I am looking at a presentation, I find it really helpful to, dissect the title and know the terms and what they mean, just so that later on I'm not confused or I'm not like, what does this even mean? And if you define the title, it kind of helps you know where the paper is going, if it's pro, if it's against, if it worked or if it didn't, what is the model system and stuff like that. So I am going to give you a little background information on sex determination pathways, climate change, sex ratio equilibrium, sex determination methods, and how they all relate to each other and how they relate to turtles. Then I'm going to talk about my study, which was done by Tasek in 2020. We're going to talk about the hypothesis, the technique that they used, the figures, and the results. And then lastly, a take-home message was just some points that I wanted you to take away from this presentation, the most important ones. So let's start off with types of sex determination. So there is two types. There is genetic sex determination, GSD, which is a process that begins after fertilization of the germ cells, and they fuse to create fertilized eggs. The germ cells carry the genetic information that's inherited from our parents. So GSD is seen in humans. And that is why we have the X and Y chromosomes, XX for female and XY for male. In the other hand, TSD is seen in alligators, crocodiles, and turtles. It's a non-genetic process. And so instead, these animals have this enzyme called aromatase that converts into hormones that determine the sex of the animal. So if you wanted to produce a female, you needed to have high levels of aromatase and produce large amounts of estrogens. And to produce a male, you would have to have low levels of aromatase and high levels of androgens. There are other factors that people can say that affect the sex of the animal, but temperature is the common link to everything. Also, temperature alters the aromatase activity. So like I said, higher means female and lower means males. So in this picture, you can see the proportion of males and the temperature that it has to have to produce males. So high temperature is for females and low temperatures for males. Yeah. So to go more into depth of the GSD process and how that works, we'll talk about the gonad. The gonad is a primary reproductive organ that determines the sex of the animal. So this is kind of like a diagram of what the gonad, the path that the gonad can take. It can take either, it can take one of two paths, to create the ovary or to create the testy. In order to create an ovary, it would have to express the proteins CIRBP and aromatase. And to produce testes, it would have to express the proteins DMRT1, COX9, and AMH. And I find it really interesting that it had like question marks. I was like, what does that mean? And I talked to one of our professors, and she said that it means that they don't know specifically what triggers that expression. Like it can take any of those paths, but there is a specific mechanism that either that's like, oh, I don't want to express DMRT1, I want to express aromatase, which is why it creates a female. So, and then I kept reading the article, and it did say that they were not sure what mechanism it was. It was something that they were looking into, but they were not sure what mechanism, which is, it's really cool, to think that one little thing, but that's just how that works. But we're not really sure what it is, and could we alter it to like, change the amount of males that can be produced? Or like in the case with the alligators, can we do something and make it so that the females can be produced more, and or female could be produced more, So what's hurting TSD species? Sea turtles face many obstacles. They have to hide from their predators, and also from us hunting them for their meat and their eggs. And also from the things that we bring to them, pollution and climate change, global warming. So climate change causes warmer weather, and warmer weather, higher temperature develops more females. So the gonad goes through the pathway of the ovary. So it skews the sex ratio, that can potentially decrease the population growth, and it can be a threat to them, like for extinction and stuff like that. It can decrease the population growth because, like I said, if from the crocodiles, if it produces a bunch of males, they won't be able to reproduce. If it produces a bunch of females, it wouldn't have, I mean, it would be it wouldn't like slow down the production, but it wouldn't be balanced. So like I said, climate change is a big factor when it comes to like the population of these animals. It skews the sex ratio of the TSD animals. It can lead to extinction if they're not able to reproduce. And only the species that can evolve to new thermal conditions have a greater chance of survival. So can sea turtles adapt to climate change? In this article, the temperature depending sex determination in sea turtles in the context of climate change, uncovering the adaptive significance. Oh my God, I'm so sorry. They did find out that sea turtles could behaviorally adapt to favorable nesting conditions. They could do that in two ways. The first one is spatial shifts, which is they are able to move to new areas to lay their eggs, or they're able to lay their eggs a certain amount of like at a time that they know that the eggs will be the eggs can successfully develop. He did say that loggerheads, which is a species that we'll talk about in our study, they do not do spatial shifts. They adapt to the thermal conditions by adjusting their thermal tolerance, which is, it's better because that means that they're evolving. Yeah, that it's a good thing, but it's not like it can be done right away. You know, it takes time for them to be able to do that, which time doesn't wait for them for anybody. Conservation. In this other study of effects on global warming on species with temperature depending sex determination, bridging the gap between empirical research and management, the author talked about relocating eggs into an in situ, which is keeping the eggs where they're at, like where their nest, where they lay their eggs, but being able to protect them and monitor them will help. If the relocation is done incorrectly, it could interfere with natural selections for the traits of turtles being able to adjust to the temperature, and just kind of if we were to keep them in a laboratory, we would just keep them, in a temperature where they will be reproducing, but then once they go to the wild, they won't be able to survive because that's not the temperature that's really in their natural habitat. So it can be bad if we don't relocate them to a place where they can evolve. And even though it's like not like this is not their fault, they're not the ones that created this, it was us, they still have to find a way to fend for themselves, and see if they're able to evolve and then pass it down to to future generations so that they can be able to be resistant to these changes that are happening. So like I said, climate change does alter the sex ratio, and we would think that sex ratios were like, I assumed that they were balanced in order for them to work, but it's actually really rare to see balanced sex ratios. They tend to be biased towards females just because, we did talk about the high temperatures producing females, and, climate change, it does change the climate and the weather to be warmer. But yeah, so in this chart, it shows you different species of turtles in different world regions and their sex ratios. We can start off with C. caretta, which is actually the loggerhead that we just talked about. This is what I love about this. A lot of these articles kind of had the same species or had the same methods, or kind of they reference each other. So that was really helpful. Because you're like, okay, well, I'm in the right path. So for this one, we can talk about the sea caretta or the loggerhead in Greece. As in the ratio there is 1 female to 1 male, and in Georgia it is, oh, that is here, North Atlantic Georgia, it's male bias. And like in Turkey is 1 female to 3 males. In Costa Rica, 20 females to 17 males. So yeah. So in order to control that, we kind of want to identify the sex of the turtles in advance. And we have found methods to do that. Previously, we have been euthanizing the turtles, which is kind of, to me, it's counterproductive, because if you are trying to save the turtles and they're endangered species, why would you euthanize them? But basically they would gather eggs from the nest, have them overdose on anesthesia, and then they would examine their gonad. I didn't find it. I didn't understand why they did that, but yeah, so they stopped doing that method because it was unethical, and they went on to do histology and laparoscopy. So histology is just like the study of the structures, a way of kind of like referencing, like, okay, well, this is what or like it's the study of a tissue. So they're basically like just comparing each other. So for like the first picture, that is a picture of the ovaries. And so that's kind of like what they have, and that's how the ovaries have to look. So they would just compare the turtle's gonads and stuff, and yeah. But they would do that with the laparoscopy, which is I'm so sorry, I have my mouse here. So the laparoscopy is a surgical procedure that examines the abdominal and the pelvic organs by doing an incision in the abdomen. So like you can see, that is a laparoscope, laparoscope. I'm sorry. And that is a turtle. So then they found this other method, which is what my study is going to be about, which is the blood immunoassay test. And they found out that that one's less invasive, and they're able to test multiple nesting sites worldwide because they're not using okay, so let's just get into this stuff before I say a lot of things, before I spoil it for you guys. So this is the study that I did. It was on neonate turtles, and they wanted to see if they were able to identify their sex based on blood samples. They did the study on both the T. scripta and the C. caretta. So the T. scripta is the red ear slider. It is a freshwater turtle. It uses its flippers. It's carnivorous. It's an endangered species, and its lifespan is 30 years. In the other hand, we have the C. caretta, which is the loggerhead. It's a marine turtle. It uses its arms and claws. It's an omnivore. It's also an endangered species, and its lifespan is 70 to 80 years. And I did not know why they emphasized like flippers and arms and claws. I was just like, what? Okay. So then I did my research, and it turns out that the arms and claws are more used by the loggerheads because it helps them with mating, and it helps them have more grip, like with sand when they're walking in the sand. On the other hand, the red ear sliders use their flippers more because they're able to swim faster with them, like to hide away from predators and stuff like that. I found them really cute. Their eyes, like the loggerhead's eyes, are so cute. They're like puppy eyes, but like 10 times bigger. So they're adorable. So what was the hypothesis? That they wanted to find this they found this technique that they hope will determine the sex of the neonate turtles. So they did the study on both the loggerhead and the red ear slider, and they got a bunch of samples of both type of turtles, and they incubated them at male producing temperature and female producing temperature to ensure that they would have, both females and males. Their technique was the immunoassay blood test, which was it tested the presence of proteins that would help determine the sex. So like we talked about the first diagram that we talked about with the gonad and the DMRT1, the COX9, the AMH, and the CIRBP and aromatase. So basically, those are the proteins that they're looking for. They're trying to see if there's like a certain protein that's present in males but not in females, or in vice versa, and if that can help them identify the sex better than the other methods that they're using. So they collected, for the red ear slider, they collected 0.2 milligrams, 0.12 milligrams of blood, and it had to be done within 1 to 2 days of them hatching. And then for the loggerheads, the same time, but it was 0.13 milligrams. So it was a small amount of blood that they were using, and they were able to identify the sex right away. So it was better, because in other cases, I mean, if they were doing the surgery where they examine your abdomen and your your cervix, and they would have to do a surgery on you, and then they would have to, well, not on you but on the turtles, and that would mean that they had to take care of them after, and what if they die and stuff like that. So it was more it was more money and more time that they had to use just to identify the sex. This is the first figure of the study, and it's not my favorite, but it's a Western blot, and it was a Western blot of the AMH protein. So this is on the red ear slider. It has two lanes, actin and AMH, and it's a side-by-side female versus male. Actin is used here as a loading control, which is why it's present in all lanes. And AMH, it stands for anti-Mullerian hormone, but you can see that it's only present on the males, right? I don't like this law. I don't like it because first off, the lanes are too big. They're thick. This one looks okay, but the next one, I'll tell you about it. But you see how when it, like for actin, okay, it's one band, but then for males and the AMH, it's all over the place. You can see a band there, but other, like, it just doesn't, it looks messy. It doesn't give us, I mean, it does give us information. It does tell us that AMH is present in males, but not in a very pleasing way. This, and then, okay, this is the other Western blot, and that is on the loggerhead, the C corretta. And it has both, it has two lanes, actin and AMH. And this is what I hate, because usually when I look at an article or like a figure, I like them to be consistent. I like them to be in the same exact spot. Every single thing is in the exact, it's just pleasing to see that. But in this case, you couldn't really get that, and those were the only figures that they gave you. But basically, the same results. Actin was present in both and all, and AMH was only present in males. But you see how on the red ear slider, they compared the female to the male, and in this one it was female male and male female. Like, it was just all over the place. But the bands still look ugly for males too. It was just all over the place. I don't know what they were thinking. I didn't like it, but it did show what they wanted to show, which was that AMH protein was present in males. So this is like an overall result of the Western blot. They did the Western blot on both, like I said, T scripta and the C corretta. The incubation treatment, the MPT is the male producing temperature, and the FPT is the female producing temperature. For the red ear slider, they had 5 males and 5 females, and for the C corretta, they had 2 males and 3 females. Then you see the different proteins, which is the same diagram, which is the same proteins that we saw in the diagram that we talked about in the beginning. The COX9, DMRT1, AMH, and I don't know if you guys remember, but COX9, DMRT1, and AMH were all in the path of the testes. And it does show you that. It shows you that out of the 5 that were produced in a male producing temperature, 5 of them had that protein. And DMRT1, AMH, and the aromatase was for female. But then you could see that in CIRBP, for both turtles, it was present in both the female and male, which indicated that it wasn't a good marker. Like, you couldn't really tell, like, if you wanted to use a protein to identify a female or male, CIRBP wouldn't be a good choice, just because the results will be inconclusive since it's present in both. This was another Western blot result, but it wasn't actually a Western blot. It was just a way of them kind of checking themselves and being like, okay, this is my method and this is what I want to do, and I'm trying to do the same thing that that laparoscopy is doing. So let me check myself and see if the results I got will match with the laparoscopy. And you could see that it did, because they did have different amounts of turtles, but the 10 turtles that they incubated at the male producing temperature actually resulted in 10 males, and 10 females produced 10 females. And for the C. corrida, 28 males and 31 females, and it did produce them, in fact. So it checked themselves, they checked themselves, and it did go as planned. So what were the results for this study? AMH plays an important role in sex differentiation, and AMH is only seen in males, which can indicate that it can be a sex-specific marker. So it can help identify females and males. Blood samples can help them do that. And it can be, and then AMH, so then AMH can be used as like a control. Like if it's present, then it means it's a male, and if it's not. But I really like this study because it was just like, it was an easier approach that they got. It was easier, it was convenient, it was better than like doing a surgery or euthanizing the turtles. And it did work. It did show that they were able to identify what they wanted to identify. So before we end this presentation, I do want you guys to go home and have these 3 points, and just know that temperature, sex differentiation, species depend on climate to reproduce. The study that we saw, TASAC, that they did in 2020, developed a method to determine the sex of neonate turtles, and they did. And the method did work. It did help determine the sex of turtles in a less invasive way by blood sampling. And the sex determination methods will assist with conservation efforts. Being able to identify the sex will help us, see what this nest will produce, if it will produce males or females. But yeah, thank you for watching.